This is Sten Nilsson. I've been asked to give a presentation on the Guadex platform and on OsteoDex. In my first part of my presentation, I will focus on Guadex and will give a number of examples showing the versatility of this methodology to produce new drug candidates and to optimize already existing drugs for better efficacy. In the second part of my presentation, I will give an overview of current treatment principles for metastatic prostate cancer, and I will also give some information about OsteoDex, our main first-in-class drug candidate. So I will start with the Guadex platform. The backbone of the platform consists of the polysaccharide dextran. As you probably know, this compound has been used as an important blood plasma expander since the 1940s. Of special interest, dextran is well suited for conjugation with other molecules. This finding has become the basis for our platform Guadex, which we have used for development of novel drug candidates. The platform was developed by Anders Holmberg, who has been working with dextran for several years. He has also had the privilege of working with Professor Björn Engelman, and he's the one who, together with Professor Anders Grönvall, developed dextran from idea to one of the most important drugs. I will now give the first part of my presentation and I'll give some more information about this Guadex platform. It's a versatile platform and, as I said, suitable for conjugation of both biologically active moieties and drugs intended for target-specific treatment. It's also useful for the modification and optimization of drugs and drug candidates, and I will soon give some examples on that. The principles of the Guadex platform consist of three important steps. The first step involves selection of the appropriate molecular weight of the dextran, which by nature is polydisperse. We can, for example, choose dextran with a medium molecular weight of 70 kilodaltons or 40 or 20 or higher. We have chosen to work mainly with dextran 40. The choice of molecular weight is determined by the area of use, that is, if you want circulation of your drug candidate mainly in the blood plasma pool, or if you need smaller molecules with the ability for renal filtration. Second step, you choose the electric charge density. That is, if you want a positively or negatively charged molecule, or if you want a neutral one. The selection of net charge is important for the intended use of the drug candidate. For oncological purposes, a positively charged anionic molecule is often preferred, since cancer cells are usually negatively charged due to the rich presence of sialic acid on the surface. The third step, a second type of molecule, is conjugated to dextran backbone with the purpose of achieving desired biological or targeting activity. Conjugation of additional molecules is sequential, that is one type of molecule at a time. The first type of molecule is here visualized as an orange-colored triangle. This stage is also including the choice of conjugation density, that is, the number of molecules per molecule of dextran that is needed. Then obviously there's a possibility of conjugating additional types of molecules, in this case visualized by blue diamond. And then there's a possibility of conjugating a third type of molecule, for example radionuclide, with the intention of delivering targeted radiotherapy to tumor tissues. The alternative is, of course, to conjugate a cytotoxic chemotherapeutic moiety. I will now present 8 different examples in which we have used the Guadex platform with a goal focused on clinical application. The first example is OsteoDex. a drug candidate intended for treatment of metastatic castration-resistant prostate cancer. That is the most serious form of the disease, where the skeleton is usually affected by metastases, and where the usual hormonal treatment is no longer sufficient to curb progression of the disease. OsteoDex is our main drug candidate. In this case, we've linked alendronate to the dextran backbone. Alendronate is used here for targeting purposes, that is to get the substance to bind specifically to metastatic areas in the skeleton. The second molecule that we have coupled is guanidine, and this is a molecule that contributes to the cell-killing effect. I'll give some more information on preclinical and clinical data towards the end of my presentation. This example of is SOMADex. We've here linked the naturally occurring inhibitory hormone somatostatin to dextran. The rationale behind this is that castration-resistant prostate cancer cells express somatostatin receptors, mainly type 1, on the cell surface. The synthetic somatostatin receptor analog lenalidomide, I think it's important to say, binds mainly to receptor number 2. And this is therefore not suitable for castration-resistant prostate cancer. Natural somatostatin, which also bind to the receptors, all 5 receptors actually, has an inhibitory effect on these cells. But unfortunately, it's not possible to use this hormone since it has much too short a half-life in the blood circulation, around 3 minutes. By constructing SOMATODEX, we have been able to extend the half-life to 27 hours. This is important for the biological effect. It binds with high affinity in the nanomolar range to all 5 receptor types. We have conducted the first study on patients, a so-called phase 1 study. SOMADex is very well tolerated and associated with only few and mild side effects. In the highest dose step, we could see objective treatment response. We saw decrease in PSA values and also pain relief, as reported by the patients. The third example is PSMA dextran. Prostate cancer cells express on the cell surface a protein, prostate-specific membrane antigen, PSMA, which is in fact an enzyme, carboxypeptidase. We have developed a drug candidate, PSMA-DEX, that binds specifically to PSMA on prostate cancer cells. PSMA-DEX is intended for target-specific treatment, that is, carrying cytotoxic substances or radionuclides to the cancer cells. Similar molecules are being tested by other research groups and also by pharmaceutical companies for theranostic purposes, that is, for diagnosis and treatment of prostate cancer. But those drug candidates are mostly monovalent, that is, they carry a single cytotoxic molecule. PSMA-DEX has the advantage with its avidity. It binds to a large number of receptors on the tumor cell. Also, it carries a high payload, that is, a large number of cytotoxic molecules per dextran molecule. And this is important, and it's an improvement over other drug candidates, as I see it, that are currently being tested. Fourth example is the versatility of the Guadex platform. The 3 examples that are already mentioned reflect this. In all types of targeted treatment, it's important to develop drug candidates who have a high avidity, that is, binding to a large number of specific receptors or a specific receptor, and at the same time carrying a large amount of the biologically active molecule. In this case, we see a great potential with our Guadex concept. Example number 5 is the comparison between Guadex and monoclonal antibodies. Antibodies are, as you know, currently being used for treatment of a large number of diseases. However, these are usually monovalent or bivalent. The antibodies can be monospecific or bispecific, that is binding a single type of target structure, or, as in the bispecific case, two different target structure on the tumor cells. With the Guadex platform, antibodies can be conjugated to the dextran chain, resulting in a molecule that can simultaneously bind a large number of specific receptors on the cell surface. This concept is important where the tumor killing effect is enhanced by cross-linking cell surface receptors. The possibility of coupling small molecules such as antibody fragments is obvious. There's also, of course, the possibility, as I've already mentioned, to conjugate a cytotoxic molecule or a radionuclide to further increase the tumor cell killing effect. Number 6 represents completely different and I think exciting possibility, that is to improve the solubility of poorly soluble drugs and biological active substances. An example of this is alendronate, which I mentioned previously. This is a bone anti-resorptive drug of the bisphosphonate type. The solubility of this drug is quite limited. But we have conjugated alendronate to dextran with using the Guadex platform technology. We were able to get the drug quite soluble. We were also able to show that the drug retains its biological activity. This we have shown in preclinical studies together with researchers at the Atlantic in Nantes, France, and with the help of Professor Ulf Lerner's research lab in Gothenburg and Umeå. We have also been able to verify this finding in our phase 1 and phase 2 trials in prostate cancer patients. We've also obtained data showing increased solubility and retained biological activity with other poorly soluble drugs such as flutamide, which is an anti-androgen, and abiraterone, which is one of the novel endocrine drugs that I will talk about later on. The 7th example is the optimization of pharmacokinetics and biodistribution. I've already mentioned that such an optimization, namely SOMADex, where we were able to link natural somatostatin to dextran with the help of the Guadex technology, and thus extend the circulation time from just a few minutes to 27 hours. Something that is important in the treatment with this hormone. As a result, natural somatostatin can be exploited for clinical use. We have, as I mentioned, data from the first phase 1 clinical trial with this drug candidate. Regarding size, by using a molecular weight of 50 kilodaltons or higher as backbone, we can make a drug candidate to remain in the bloodstream, in contrast to lower molecular weights, where the molecule is supposed to be filtered through the kidneys. We also have clinical data from intravesical treatment, where we have conducted a pilot study on patients with superficial bladder cancer. By using cationic backbone, we could see quite a good accumulation in superficial bladder cancer compared to normal bladder mucosa. So we intend to further develop the concept for superficial bladder cancer, where there is a really big need for new and active drugs. The 8th example is the potent cell-killing effect of OsteoDex in other types of malignant diseases. We have preclinical data showing good cell-killing effect on breast cancer cells, even those that are so-called triple. Data from in vitro studies have also shown good cell-killing effect in lung cancer. OsteoDex inhibits a strong effect also on myeloma cells, and the combined effect on tumor cells and at the same time on osteoclasts is important, especially in this disease, multiple myeloma. So in summary, the Guadex platform can be used to advantage as a carrier molecule for drugs, for growth hormones, antibodies, antibody fragments, receptor inhibitors, and also antibacterial and antiviral substances. We have our own experience from coupling guanidine and alendronate, as in the case of OsteoDex. Coupling of alendronate, somatostatin, PSMA binding molecules, and other molecules are examples where OsteoDex and alendronate and somatostatin have been tested in phase 1 and phase 2 clinical trials. The possibility of conjugating poorly soluble drugs is of course of interest and can open the door to increased availability and better utilization of such. We have, as I mentioned, conducted early clinical trials on 4 of our substances, which are based on the Guadex platform. The versatility with the Guadex platform lies in being able to change important variables such as molecular weight, net electric charge, half-life in the blood circulation. Furthermore, the platform allows selection of appropriate avidity and properties that are associated with penetration of, for example, the cell membrane and the membrane of the cell nucleus and mitochondria, etc. The platform allows the manufacture of drug candidates that are particularly suitable for intravenous administration and for intracavitary administration, as in the example of intravesical treatment of superficial bladder cancer. So in conclusion, although we have so far focused on the development of mainly oncological treatment, it's important to convey the message that the platform is, for obvious reasons, very well suited for non-oncological treatment. For example, antibacterial and antiviral treatment. I will now focus my presentation on metastatic prostate cancer. In this context, I will mention OsteoDex and the intended place for this new drug candidate in the treatment arsenal. I will also point out a paradox here that the need for new drugs has actually increased significantly in the last 2-3 years, despite the advent of new medicines. Now, briefly go through the modern treatment principles for prostate cancer. A localized cancer, that is one that has not yet spread, can today successfully be cured with radiation therapy or surgery, a combination of these. The least aggressive forms of prostate cancer can today be just monitored without curative therapy. We call this active monitoring, just controlling the disease. And with the most aggressive forms is that they can progress, especially if they have been already spread at the time of curative treatment. You can usually see this in the form of a PSA relapse. If the prostate cancer has already metastasized, there are today a number of medical treatments that can slow down the disease. But however, none of these treatments can cure the disease. I usually tell my patients that one has to live with the disease, check it regularly, and make appropriate therapy changes as needed. Prostate cancer metastases often settle in the skeleton, here visualized with the bone scintigraphy, and the metastases are seen as black spots in this scintigram. Evidence-based treatment in this situation is hormonal therapy, where the goal is to prevent testosterone from stimulating the cells, or to be more precisely the dihydrotestosterone, which is the fuel for prostate cancer cells. The treatment is usually done medically with regular injections of so-called LRH analogs, Such a drug prevents the production of testosterone. We can also use drugs that block the effect of dihydrotestosterone on the tumor cells. The disease responds very well in majority of cases to hormonal treatment. The treatment effect can, in some patients, last for several months and even years, while the treatment effect in other patients lasts only for a few months or even less. Most patients, some of the tumor cells change over time in that they become increasingly insensitive to the usual hormonal treatment. The tumor cells have become castration resistant, CRPC. There are several ways for the tumor cells to develop castration resistance, that is, to continue the growth and metastatic ability despite hormonal therapy. I will not go into detail in all of these, just listed here just a few of them. There's a number of important drugs available today for castration-resistant prostate cancer, and all of these have been shown to curb disease progression, relieve symptoms, and prolong overall survival. The first of these important drugs is docetaxel, which is a cytotoxic drug, chemotherapy. The second drug is Provenge, sipuleucel T, an immune treatment. This concept has not been widely used for various reasons. The third drug is Zytiga, abiraterone. This is a hormonally active drug given orally. It inhibits the tumor cell's own production of androgen. It's interesting that the castration-resistant cells can develop their own production of androgen or testosterone. That is their own fuel by an enzyme called CYP17. Abiraterone inhibits this androgen production in the tumor cell and thus slows down the cancer growth. The effect is usually transient, but can in many patients prove effective over many months and in some cases even years. The next drug approved for castration-resistant prostate cancer is enzalutamide or Xtandi. Also, this is an oral drug that is hormonally active. It inhibits the effect of the dihydrotestosterone on the so-called androgen receptor and thus curbs disease progression, relieves symptoms, and also this drug prolongs overall survival. And as in the case of abiraterone, this effect is usually transient, but can in many patients prove effective over many months and also in some patients even years. The problem is that the disease usually eventually progresses on docetaxel, abiraterone, or enzalutamide. So therefore, a second line of therapy must be considered. Paclitaxel or Yevtana is a chemotherapeutic drug that, like docetaxel, is given intravenously at 3-week intervals. It's approved for treatment of patients whose prostate cancer has failed docetaxel, progressed on docetaxel. Abiraterone and enzalutamide can be used as second-line treatment provided they have not been used earlier in the course of the disease. Radium-223 or Xofigo is an alpha-emitting radionuclide, which is used for target-specific treatment of bone metastasis in prostate cancer. The drug is given intravenously on an outpatient basis every 4 weeks, totally 6 treatment cycles. This is one of the new drugs that can curb disease and prolong survival while improving quality of life. I've had the privilege of being involved for many years in the development of this, in the clinical development of this drug, which has now been given to more than 50,000 patients globally. PARP inhibitors represent another type of drugs. They inhibit an enzyme poly-ADP ribose polymerase, which is involved in the repair of DNA damages. One example of such a PARP inhibitor is olaparib. The treatment presupposes defective genes that are involved in DNA repair. Unfortunately, the proportion of prostate cancer patients who can benefit from this particular treatment is quite low. Immunotherapy has become an important treatment concept for a number of malignant diseases. The concept is here represented by the PD-1 inhibitor pembrolizumab. Unfortunately, only a small portion of prostate cancer patients benefit from treatment with this drug and this immunological approach. Significant progress has been made combining castration treatment with chemotherapy or one of the novel endocrine drugs already at the time of detection of metastatic disease. This is an important paradigm shift in the treatment of prostate cancer. The first drug that has brought success in the form of overall survival benefit is docetaxel, which we have already discussed earlier in this presentation. The other three drugs that have successfully improved treatment outcomes when combined with castration therapy are abiraterone, enzalutamide, and apalutamide. A not unusual situation is that a prostate cancer can develop castration-resistant disease and still be localized. That is, it has not yet metastasized. These patients may have a prostate cancer that for some reason has not been treated or simply could not be treated with a curative intent. These may also be patients who have relapsed after surgery and/or radiation therapy and who have subsequently undergone hormonal treatment and progressed into a castration-resistant stage. A treatment strategy that has proven successful is to combine castration with one of the novel endocrine drugs in non-metastatic castration-resistant disease. Here in the form of apalutamide, enzalutamide, and darolutamide. All three represent the new generation of androgen receptor inhibitors. Paradox is that there is now actually an increased need for new drugs for prostate cancer. We can see that quite a few of the new drugs are used much earlier in the disease continuum than before, which is a good thing. The problem is that you have an extremely limited effect of these drugs if you try to use them again later on in the course of the disease. So we are thus in a big and growing need for new active drugs, and that these new drugs also must have a unique mode of action. We can now see that many drugs for castration-resistant prostate cancer simply fall away since they have already been used in the early course of the disease. The paradox is thus that the need for new drugs for castration-resistant prostate cancer has increased in recent years, despite the advent of very active new drugs for prostate cancer. Here I've tried to describe the important drug change in their place from second-line therapy to first-line therapy. We thus have a big need for completely new types of drugs to fill the unmet need. It's in this area that we work with our new drug candidate, OsteoDex. First, some additional important words about bone metastatic castration-resistant prostate cancer. Almost all patients have bone metastasis already in the early course of the disease, before other metastases, which are so-called visual metastases, before these are formed. The biological process, which we call the vicious cycle, takes place in bone metastases. It involves an interaction between the tumor cells and the normal cells of the skeleton Tumor cells that spread from prostate cancer very often go to the skeleton. The reason for this is not completely clear, but it can be explained by the seed and soil theory that the breeding ground for metastatic cells is optimal here. The skeleton has normal cells that participate in the physiological turnover to keep the skeleton healthy and strong. There are cells that resorb the skeleton, the osteoclasts here in the red color, and other cells that participate in the physiological bone formation, the osteoblasts, down to the left in the figure. These two cell types are in a well-balanced equilibrium with each other in the healthy skeleton. The big problem arises when metastatic prostate cancer cells settle in the skeleton. The tumor cells are stimulated by a large number of growth factors that are formed by the normal bone-resorbing osteoclasts. The tumor cells in turn stimulate the normal bone-forming cells, the osteoblasts, which in turn stimulate the bone-resorbing osteoclasts. This vicious cycle is unfortunately beneficial for the tumor cells, which by this vicious cycle can continue to grow, to change character, and become even more aggressive, and to further metastasize. First to other parts in the skeleton, and then to extraskeletal visual organs. The vicious cycle outlined here is of course much more complex and involves other normal supporting cells in the skeleton and cells involved in the immune system. Many new drugs are explored to further improve the inhibition of the vicious cycle in bone. As I mentioned, the skeleton is the main and most important metastasis site for prostate cancer. These metastases often spread to new parts of the skeleton, and they also change their character to even more aggressive phenotype before spreading to other organs. An important reason for treating bone metastases as effectively as possible is the fact that the most common cause of death in prostate cancer is due to the skeletal metastasis and the bone metastasis related complications such as fractures and spinal cord compressions, etc. We have since a long time been working with and further developed such drug candidates with intention to inhibit the vicious cycle and, as a consequence, the metastatic disease as such. Our main drug candidate is OsteoDex. We have data from both preclinical and clinical studies showing that OsteoDex can effectively inhibit the vicious cycle. We know, as I said, that the disease negatively affects the skeleton with so-called symptomatic skeletal events, that is, pathological bone fractures. But another problem that we have with many other treatments is, for example, not least the hormonal ones, is that also these have a negative effect on the skeleton with osteopenia and osteoporosis. So one of the advantages with OsteoDex, in addition to disease control, is its bone restoring effect. This is something that we have seen not only in preclinical experiments, but also by studying bone markers in the blood of prostate cancer patients treated with OsteoDex. The OsteoDex molecule has a unique mode of action and is thus by definition a first in-class drug candidate. It basically consists of dextrane molecule as the backbone, as I mentioned. And dextrane is well-known drug, also as we have mentioned. We have conjugated alendronate to dextrane, and alendronate, which is a bisphosphonate and a bone-preserving agent, usually used for osteoporosis, has here, in our case, been specifically used for specifically target OsteoDex to bone metastasis by binding to hydroxyapatite, which is always exposed. Sorry about that. In the areas where the vicious cycle is active. To the dextran molecule, we have also attached another molecule, the guanidine, and this is the moiety that gives OsteoDex its cell-killing cytotoxic activity. We have performed a number of preclinical studies. This image shows the effect of OsteoDex on in vitro cultured human prostate cancer cells, that is, cells that we have grown in the laboratory. On the y-axis, we have the number of cells, and on the x-axis, the time in days. The red curve shows that untreated cells grow relatively quickly over time and eventually cover the bottom of the so-called petri dishes. Cells that are subjected to treatment with OsteoDex do not grow, as shown by the green curve and the blue curve. This cell line does not respond to hormonal treatment, but still we have a good effect of OsteoDex. We have been able to show the same cell-killing effect also with other cell lines derived from prostate cancer. One of the major clinical problems with prostate cancer is that tumor cells usually change the character over time and become therapy resistant. This is one of the major reasons for the urgent need of new drugs with different modes of actions in this disease. We have access also to cell lines that are resistant to the novel endocrine drugs. Here's an example from such a cell line. The effect of OsteoDex is seen in the figure to the left. The y-axis shows the percentage of surviving cells, and the x-axis show increasing concentrations of OsteoDex. The cells, which were treated for 72 hours, decrease with increasing concentration of OsteoDex. At a concentration of 4 micromoles, only 50% of the cells remain. The image to the right shows the effect, or rather the absence of effect, when treated with one of the novel endocrine drugs, apalutamide. Prostate cancer also usually develops resistance to docetaxel, the chemotherapeutic drug, which is one of the gold standards in this treatment. We have developed a docetaxel-resistant cell line by treating it for a very long time with docetaxel in low concentration. In the end, we have developed a cell line that is completely resistant to treatment with docetaxel. The figure to the right shows the absence of the effect of docetaxel, while the left figure shows that OsteoDex maintains its high cell-killing effect. We have, as I mentioned earlier, also studied the effect of OsteoDex in vivo. These experiments were done at the Atlantic Bone Screen in Nantes, France. The results from this preclinical model showed that tumor growth was inhibited. We could also see reduced tumor volumes and evidence of bone repair. OsteoDex has also been studied in patients with bone metastatic castration-resistant prostate cancer. The first study, the so-called phase 1 clinical trial, was performed with increasing doses of OsteoDex. 4 patients per dose step, 7 dose steps in total. The primary objective was to study safety. The results show that OsteoDex is very well tolerated, even in the highest dose step. And in the highest dose step, objective responses were seen with reduction in the bone markers. Subsequently, we performed a randomized 3-armed double-blind study. Patients were randomized to one of 3 different doses of OsteoDex, so all patients got active treatment. By double-blind, this meant that neither the patient nor the study center was aware of the dose given. The treatment, which was given intravenously and on outpatient basis, was repeated twice a month, a total of 10 treatment cycles. Thus, all patients received the active treatment with our drug candidate, OsteoDex. We saw no difference between the 3 doses regarding efficacy, and this was actually expected, since even the lower doses should be fully sufficient to saturate the metastatic areas of the skeleton with our bone-specific substance. So we interpret this as a good result. Another good finding is that none of the patients suffered serious side effects from the treatment, even in the highest dose. The conclusion is therefore that the safety with OsteoDex is very good. The drug was also very well tolerated with only some joint and muscle pain, which could be managed with analgesics. By using the technique Bone Scan Index, BSI, we could see that metastasis decreased in distribution and activity and size, and that the effect that we saw with OsteoDex was in the same range, size, I would say it was on par in the same size range as docetaxel, abiraterone, and enzalutamide. And we were also able to see an equally good effect in patients who had previously been treated with and progressed on novel drugs such as docetaxel, cabazitaxel, abiraterone, enzalutamide, and radium-223. This is in line with the fact that OsteoDex has a completely unique mode of action. We interpret this as a good result. As I mentioned earlier, OsteoDex interferes with the vicious cycle in bone metastatic prostate cancer by inhibiting the tumor growth as well as the bone resorbing cells, the osteoclasts. This figure shows the effect on osteoclasts in the patients who completed the treatment of 10 cycles. The vast majority of patients showed a robust decrease in CTX in the blood, visualized in this waterfall plot. CTX is an important marker for the osteoclast activity, which reflects the activity of the metastatic process, that is, the vicious cycle of this disease. As I mentioned, the low doses showed as good a disease inhibiting effect as the highest. All patients received, as I said, active treatment with OsteoDex. The follow-up extended to 2 years after the end of treatment. The patients who showed a clinically positive effect in the form of stable disease and/or tumor reduction had a significantly longer survival compared to the patients who did not respond to the treatment. Complete data from the study will be compiled during the year and then submitted to a scientific journal for publication. So to summarize, OsteoDex has a unique mode of action compared to all known drugs and has shown effective cell killing in preclinical studies, effectively kills docetaxel resistant cells, just as it also kills cells that are resistant to the novel endocrine drugs. It effectively inhibits the vicious cycle in bone metastasis, and we have data from preclinical studies verifying the bone-forming activity of OsteoDex. This is a finding that is supported from the analysis of bone markers in patients who were treated with OsteoDex. OsteoDex has a unique mode of action and thus also meets the definition of a so-called first-in-class drug candidate. The drug candidate was very well tolerated in both clinical trials and did not show any treatment-related serious events, nor did it result in treatment due to side effects. Type of quantitative bone metastatic burden, we were able to see treatment responses that were completely on par with what has been shown with other novel drugs for this disease. And it's noteworthy also to see that we saw treatment responses in patients who had already received and progressed on one or more drugs before OsteoDex. We saw equally good treatment responses in all 3 treatment arms of the study, and we interpret this as a good result. And there are patients who responded to the treatment with no progression and/or reduction of the tumor burden also showed a better survival. So back to the question regarding the place for OsteoDex in the treatment arsenal of castration-resistant prostate cancer. As we have As we already know, none of the drugs available today have the potential to cure the disease. I think that's important to state. We also have the paradoxical fact that the need for new drugs in castration-resistant prostate cancer has actually increased over the last 2-3 years, and that the reason for this is that the majority of existing CRPC drugs are now given early in the course of the disease. My very personal view is that we cannot see or expect any new versions of hormonally active drugs. The new hormonally active drugs, abiraterone and enzalutamide, apalutamide, darolutamide, they do have significant cross-resistance. So we are thus facing the fact that novel drugs with unique mode of action will be needed, and above all, that these new drugs also have a benign safety profile and are very well tolerated, and with ability, of course, to be used for combination therapies. So back to the figure with existing therapeutic options. As we see from this, the increased need for new drugs has risen in castration-resistant form of prostate cancer. My view is that a drug candidate like OsteoDex it fits very well here. This is in view of the fact that the mode of action is unique. It is completely different from current drugs. The treatment effect is seen also in patients who have failed previous treatments with one or more drugs, and that OsteoDex is very well tolerated with only few and mild side effects. A not entirely uncommon question is whether OsteoDex will compete with other drugs or vice versa. And the answer to that question to me is very simple. As long as we have no drug that can cure the disease, it is by definition so that all new efficient drugs with novel modes of action and good tolerability are indeed needed and requested both by treating physician and also, of course, by the patient himself. So I hope that this presentation of the Guad has increased, you have got to increase knowledge about the Guadex platform for development of new drugs and drug candidates. One of our main areas is prostate cancer, as you heard, and this is also the area where we have come furthest with the OsteoDex. The Guadex platform can, as I mentioned, also be applied to modify and improve the properties of existing drugs, both for malignant disease and also for non-cancer. Of particular interest to non-oncology areas is the development of antibacterial and antiviral drugs. We have already some interesting preliminary data here on these non-oncological applications, and hope to come back to these sometime in the near future. So thank you for taking your time to listen to my presentation on Guadex and OsteoDex. Thank you.